Hi, everyone. It's a pleasure to be here today. I'm really excited to talk about Synthego and how we're accelerating cell and gene therapies. And I'm Rebecca Nugent. So we look at the next generation of therapeutics coming online, and we know that there's a lot of competition in this space. First, in the competition of the number of developers that are coming, the investment that's been had, but then also from a manufacturing standpoint. There's a manufacturing shortage. We're all familiar with the supply chain issues that we face today, as well as the cost of goods for these therapies. At the same time, we know that regenerative medicine, ATMPs, are growing. There are more products coming on the market. And gene editing, and specifically, continues to advance as a therapeutic modality since its initial gene editing trial in 2019. Currently, there are 41 trials ongoing in gene editing, with 80% of them using CRISPR, or Cas9, as its nuclease. So a key problem for gene therapy developers that we see is how do they balance the throughput and speed for developing these editing approaches while keeping their eye on downstream clinical compatibility and GMP manufacturing. So at Synthego, what we do is that we industrialize CRISPR for accelerated discovery to clinic, and we do this through multiple platforms that I'll get into during this presentation. We really aim to standardize CRISPR to bring it to a larger market, and we do this to eliminate experimental variability with the newest gold standard in CRISPR cells, part of our engineering cells platform. We scale with confidence, so as you'll see in the slides coming up, we really believe in accelerating experiments through a combination of machine learning and automation to really scale the number of experiments that we do at once and then pass it along to our customers and our partners. And we're continuously improving, right? So we improve CRISPR for higher predictability and for project success. And that allows for our researchers to de-risk their projects, knowing that the edits they're making will be successful and they can test the biology. So here's a timeline of Synthego. We were founded in 2012. In 2016, we launched our first product, which was a synthetic guide RNA, or sgRNA, which you'll see across the slides, our kit. So we launched that in 2016. It became the gold standard for genome editing, and it reduced the cost and wait times for synthetic RNA by 5x. We followed that up. We looked at the market and said, what are people using CRISPR guides for engineering cells? So we followed that up in 2018 with our engineered cell lines, which gave researchers unprecedented access to engineered cells. So instead of needing to have the ability and the expertise and build out the labs to engineer your own cells, now you can come to Synthego as a commodity, just order it through the platform. At the same time, we've recognized the need for increased the power of CRISPR and how it can really be used to leverage and understand disease biology. So we launched our our arrayed screening libraries, which is a library of synthetic guide RNAs to knock out a variety of targets in human libraries, and it really enabled screening in clinically relevant cells. In 2019, we launched our high-throughput iPS-D cell line editing, so our CRISPR-edited iPSC cells. And then moving into today, we've moved into GMP manufacturing of the synthetic guide RNAs. And again, this is to accelerate research from discovery into the clinic. So this is our modular approach to biology. It starts with our different platforms. So the base is our synthetic guided RNA. We have our engineered cells, and we're moving into being a cell and gene therapy accelerator. So at Synthego, we've really leveraged machine learning with automation and gene editing to tackle the complexity of biology with engineering rigor to really build platforms to study science at scale. So I'll start by describing our HALO platform, which is what we call our synthetic guide RNA platform. Again, a theme that you should be seeing in this presentation is that we scale from research use only to the clinic, so from discovery to clinic. And we offer high-quality sgRNA at all different scales. So there's our RUO, or research use only. It's ideal for CRISPR-based studies in the research stage. You'll see to the left our HALO offerings, starting with our guide RNA kits, our arrayed libraries, as well as advanced RNA. So let's say you have your favorite nuclease, you have your favorite RNA sequence, you have a modality or a chemical modification that you need to make. We can talk to our scientists, and we can synthesize your RNA your way. Now, moving from RUO into GMP and GMP-like, GMP-like is kind of our gateway into GMP. It's ideal for CRISPR-based studies that require higher quality sgRNA with limited documentation, and that moves into our GMP, which is high-scale, high-quality sgRNA, meeting good manufacturing practice or GMP requirements. So I'm a scientist. I love to show data. This is one of 2 bar charts that you'll see in my presentation, which is you can have confidence with CRISPR editing using Synthego's guides. So the experiment to the right compared our research-only screening library guides to other commercial vendors. And the bar chart, the height of those bar charts, really shows the editing efficiency. And here we're measuring the percentage of a gene knockout or the indel frequency that was created. You can see based on the different vendors, each bar here represents a different gene that was knocked out, so a different guide that was used to knock out our target. There was variability in terms of genes to genes, but then also from vendor to vendor. And what I'd like for you to take home here is that Synthego's modified guide RNA and design capabilities resulted in higher editing efficiency. So it's not just the quality of the RNA, but it's how do you predict the guide, how efficiently will that guide cut to create your desired edit, and in this case, it's an indel. So moving on, so our core platform is synthetic guide RNA, or our HALO platform. How do we use that to engineered cells in-house? Well, Synthego makes industrialized cells a reality. We have an automation-driven parallel process where it allows us to do simultaneous editing of multiple targets at once, but also not just one cell, one edit, but we can do multiple cells at one time. So we really increase the throughput, and we, with that, decrease the turnaround time and decrease the cost. We're fueled by our technology, we're fueled by our HALO program, so our sgRNA. We take that technology and we really push this into our platform to yield high editing efficiency. And again, this all leads to rapid iterations. We have a number of product lines from immortalized cells to iPSCs. We do knockouts, knock-ins, pools, clones, et cetera. So what I'm showing here, the figure to the right, is editing optimization within multiple cell lines. All of the targets that you see here, it's the same locus, and we've just iterated the editing variables. So I hope the key thing that you look at this slide is that some cells are really easy to edit, and you can see that by HEC293. You have really tall bars, which means that we were able to get really nice knockouts with not a lot of iteration necessary. Some cell lines are not so easy. So anyone who's ever done genome editing knows that sometimes it goes great and sometimes it doesn't. What we do at Synthego is we iterate on it. We have experience through our customer cell lines that we've taken in, and then also some of the primary cells that we've worked in-house to really understand how we can iterate on these editing variables to get increased knockout percentage editing efficiency. So we pair RNA synthesis and then our automated cell engineering for tuning the optimal editing efficiency across multiple cell types and target locuses as well. So now I'll get to my favorite part of the presentation, which is how do we bridge these 2 capabilities to really accelerate cell and gene therapies. We've heard a lot of needs from the space. How do you get us to the clinic fastest? How do you de-risk this process? How do you develop a robust gene editing approach that can be consistently replicated? And at Synthego, we aim to solve 3 major challenges as designated by that triangle on the screen. The first is the technical challenge, and what we do is we bring our high throughput editing and optimization skills, we pair that with our novel chemistry to really tailor solutions for our partners. We have a regulatory component, which is key, especially in early discovery stages, to understand how do you go from research to the clinic. We can do that through our GMP guide, so understanding what sequences synthesize well, and then also through safety profiles. Some guides may cut really, really well, but if they cut other targets that are off targets, that's not ideal for a therapeutic. Now, in how we solve the economic challenges is that we can really, through this process that we've had, we can do process de-risking, and then we can also bring speed. So you go faster, you fail faster, you succeed faster, and all of this brings to more favorable economics. So one thing that I love to say is that focus on your strengths. Every company here that we've heard presenting, they all have very specific strengths, and Synthego's the same. So building on Synthego's core competency of RNA synthesis and genome engineering, our therapeutic ecosystem can create bespoke development streams to seamlessly tackle those challenges that I just talked about, technical, regulatory, and economic challenges. It reduces your timeline, so our high throughput workflows and our proprietary machine learning automation can reduce the time it takes for you to develop your strategy, and it can de-risk future failures. Do R&D with a clinical perspective, so an integrated research use only and GMP reagent stream, so you know that if something's getting produced in the REO stage, it'll scale up to GMP. And data packages to help build up your pre-IND filing. And then tailored solutions. Synthego has a number of proprietary technology that may not be included in our commercial offerings. And so through these bespoke development streams, there's technical solutions that can be developed specifically for the target and the cell type. And then we can apply some of the IP that we have within Synthego. So how does this relate, this development streams that I've talked about, in terms of our commercial offerings? So if we think about the cycle it takes to go from an idea to the clinic, there starts from target identification through GMP guide validation, all the way to approval and production. On the bottom are Synthego's current commercial offerings, which I've highlighted in our previous slides, through Halo, which is our synthetic guide RNA platform, as well as Eclipse, which is our engineered cells. And our development streams are a white glove path to de-risk those pipelines in early discovery stages. So there are 3 different development streams, starting with the lowest, which is therapeutic optimization. Let's say you're a developer, you have an approach, it may be slow, it may be clumsy. You might want to iterate on it to improve whatever features are important to you. What we can do as a partner is come in, assess your strategy, assess your cell type, iterate, develop, and then move through a clinical safety profile on and off targets, and then GMP-like manufacturing to ensure that it'll scale up appropriately. And at the same time, keeping research with an eye on the clinic, develop some regulatory documentation that you can take to the FDA or EMA for documentation of this approach. Sometimes developers may know what target they want to go after, but they don't know where to start. And that's where our therapeutic editing design comes in. We work together to identify strategies that may include different nucleases, different modifications, different guide chemistry, which I'll go into the next slide. Again, we go through that process, we do our high throughput screening, our automation, we use the machine learning and all of the tricks that we have in-house to really create the best type of editing approach. Again, going through GMP-like manufacturing to make sure that it scales up and de-risk the whole process. And then farther upstream is target optimization. We have lots of abilities in-house to do high throughput screens, guide screens, small molecule screens, et cetera, and then it goes through that pipeline. So one innovation that I'll highlight before the talk ends is CRISPR-off for precision engineering. We know that on-target editing effects are very important, so accuracy when it comes to genome engineering is incredibly important. So our scientists have created a chemically modified Cas9 guide that we're calling CRISPR-off. And so these CRISPR-off guides are inactivated in the presence of light. And so when we translate this to something that has clinical relevance, we know that if you deliver Cas9 to the cell, the longer that it's there, the more cutting it will do. Fantastic, but the more cutting it will do at your desired target site as well as your off-target sites. So with CRISPR-off, we can go in, modify, have CRISPR cut your cells at the target site, and then at a determined time, turn it off so it doesn't cut at undesired sites. That way, only the edited product at the desired locus is returned back to your patient. So you've heard me say research with an eye on the clinic multiple times in this presentation. Gene editing trials are upcoming. I mean, we've cited ARM multiple times in the annual report in this presentation. CRISPR and other nucleases are a modality that's very much coming in with gene editing and gene editing therapies. Synthego recently closed our Series E, and with that increased investment, we're really aiming to help researchers scale and simplify their translation of new discoveries. And so with a minute left to go, I'll say thank you so much for your attention. We're really excited to make therapies accessible by supporting discovery to the clinic. We're really looking for partners and how we can enable your work. This is something that we're incredibly passionate about. I'd love to talk to you. Take a screenshot of this picture of the screen right now. Contact me because I'd love to talk. And if not, hope we get a chance to talk later today at the rest of the conference. And thank you so much for your attention.